Now that we have talked about the concept of protein synthesis, transcription, and translation, we go into a very beautiful logic called as the operon. A gene is allowed to undergo transcription translation or not allowed to undergo transcription translation. So a gene will express itself or its expression is prevented with the help of a system called as an operon. So, what exactly is an operon? We take it in a beautiful way of understanding now. What is gene expression and gene regulation? A living cell has a gene which expresses itself to form either a structural protein or an enzyme. So, a gene will undergo transcription to form an mRNA. mRNA will form a protein. It could form structural proteins like keratin, actin, myosin, etc. Or enzymes, which are chemically proteins, are produced. That control whether a gene should express itself to form a protein or not express itself, so no protein is formed, is regulated. And that regulation is done at the transcriptional level when DNA forms the primary transcript or the mRNA. तो ट्रांसक्रिप्शनल लेवल पे कंट्रोल किया जा सकता है और at processing level conversion of DNA into hnRNA. And when hnRNA is processed, that is, splicing is done to form mRNA, तो hnRNA to mRNA के बीच में कंट्रोल किया जा सकता है और transport of mRNA from the nucleus to the cytoplasm. When mRNA is transported from the nucleus to the cytoplasm, तब उसका कंट्रोल किया जा सकता है. Or translational level, when protein synthesis takes place, at that time the control can be done. So you have the control of either a gene expressing itself or not expressing itself done by this mechanism. Usually, extracellular or intracellular metabolites trigger the initiation or inhibition of a gene expression. Very important statement. Extracellular or intracellular metabolites. There are chemicals which are either externally coming into a cell or present within a cell. They will trigger whether a gene should initiate its expression or inhibit its expression. तो एक जीन का expression to form an mRNA or hnRNA is regulated by a metabolite. A metabolite which is extracellular or intracellular will allow a gene expression or will inhibit a gene expression, and that is done by metabolites intracellular or extracellular, and that will control gene expression. A cluster of genes with related function is called as an operon. एक क्लस्टर ऑफ जीन्स होता है, उसे operon कहते हैं. And operons usually transcribe a single mRNA. एक सिंगल mRNA को एक क्लस्टर ऑफ जीन्स एक्सप्रेस करते हैं. In E. coli, there are some 260 genes which are grouped into 75 different operons. So, a cluster of genes hota hai which is called as an operon. and a cluster of genes. Say here there are one, two, three, four, five, and six genes, which is called as an operon. And that operon will produce a single mRNA. A single mRNA is expressed from that operon. ऐसे Escherichia coli में 260 genes are grouped into 75 different operons. E. coli, 260 genes are grouped into 75 different operons. For example, you have operons called Lac Operon, lactose operon, tryptophan operon, ara operon, his operon, val operon. ऐसे अलग अलग टाइप के operons है which are clusters of genes. Of all these operons, the most famous one is the Lac operon or lactose operon, and we will study that. But first, what is the structure of an operon? An operon is a unit of gene expression and regulation. Operon controls or includes structural genes, and along with structural genes, genes called promoters, operators, and repressor. तो जो मेन जीन होता है, the main gene which will produce an mRNA is called as a structural gene. एंड इस जीन को कंट्रोल करने के लिए देर आर जीन्स कॉल्ड प्रमोटर्स, देर आर जीन्स कॉल्ड ऑपरेटर्स एंड जीन्स कॉल्ड रिप्रेसर. द structural gene code for a protein. Structural gene forms mRNA. mRNA forms the protein. The promoter gene are sequences in the DNA that start RNA synthesis. So, promoter gene gives a message to the structural gene to form an mRNA, which will then undergo translation to form a protein. So, structural genes are controlled by the promoter genes. And promoter gene is the site where RNA polymerase attaches during transcription. So, समझो एक structural gene है. This is the structural gene, and structural gene के ऊपर बाजू में होता है promoter gene, और promoter gene के ऊपर stick हो जाता है the enzyme called as RNA polymerase. And RNA polymerase, we remember, will go from the promoter gene to the structural gene, and RNA polymerase helps in transcription of the gene to form an RNA called as the mRNA. तो mRNA बनता है स्ट्रक्चरल जीन की मदद से, बट उसको ट्रांसक्राइब करने के लिए we need enzyme called RNA polymerase that is attached to the gene called the promoter gene. So the P promoter gene is where RNA polymerase gets attached. Then we have operator gene, which are between the promoter and the structural genes. Operator gene is between the promoter and the structural gene. So here we have O gene, operator gene. And operator gene is present between the P, the promoter gene, and the structural genes. So promoter gene और structural gene के बीच में होता है operator gene. And then we have something called as the regulatory genes, which are responsible for formation of repressor which interact with operators. The repressor protein binds to the operator region of the operon. Sir, ये है promoter gene, और promoter gene के बीचों बीच होता है I gene. I gene, which is called as the regulatory gene. And regulatory gene also called as the repressor gene. Repressor gene. And repressor gene क्या करेगा? This is your repressor gene. It undergoes transcription to form an RNA called the repressor mRNA, which undergoes translation to form a protein called the repressor protein. And repressor protein goes and binds to the operator gene. repressor protein operator gene को bind कर देगा. So, which are the different genes? You have structural genes which form the mRNA. mRNA is formed by transcription, which forms proteins, enzymes. And then we have promoter gene, and the promoter gene is the one to which RNA polymerase gets attached, which will pass from the promoter gene to the structural genes and help in formation of the mRNA. You have operator gene present in between the promoter gene and the structural gene, and you have the I gene, the regulatory gene, which will undergo transcription, translation to form repressor mRNA, repressor protein, and the repressor protein will go and bind to the operator gene. So these are the different genes which are there in the Lac Operon, in the operon, of which we have this very famous Lac Operon model, which was given by Jacob and Monod. And Jacob and Monod proposed this classical model of Lac Operon, which explains gene expression and gene regulation in E. coli. एस्केरिचा कोलाई में लैक ऑपेरॉन और लैक्टोज ऑपेरॉन is beautifully represented by Jacob and Monod. Now, what is the logic of Lac Operon? So, Lac Operon is this. So, you have a cluster of 6 genes which are present in the DNA of E. coli. So, समझो this is DNA of E. coli attached to the cell membrane, and इसके अंदर you have different operons. So you have trip operon, you have his operon, you have ala operon, you have val operon. तो ऐसे अलग अलग operons है. If you remember, there are 75 different operons. This operon consisting of 6 genes is the Lac operon. So ये है Lac operon. And Lac operon में 3 अलग अलग प्रोटीन्स बनते हैं, namely permease, transacetylase and beta galactosidase. Permease, transacetylase and beta galactosidase. Permease will allow permeation of lactose into a cell. तो सेल के अंदर लैक्टोज की एंट्री होती है. There are gates, and through the gates of the cell membrane, lactose is allowed to enter. The gates are usually closed. तो gates बंद होते हैं. It is permease which will go to the gate and open the gate. Gate will be opened, and as the gate opens, lactose will enter into the cell through the opening of the gate done by permease. So gate is opened by permease. Gate is opened by permease and lactose enters inside a cell. Once lactose enters inside the cell, it is this enzyme called beta galactosidase, which will break down lactose. Lactose is a disaccharide, and disaccharide cannot undergo glycolysis. If you remember, respiration किया है हमने. Who produces energy? Glucose. Glucose produces energy, and lactose is a disaccharide. So once permease allows lactose to enter in the cell, beta galactosidase will break down lactose by hydrolysis into glucose and galactose. So lactose breaks down, beta galactosidase breaks down lactose into glucose and galactose, of which glucose undergoes glycolysis to produce energy, ATPs for the E. coli bacteria. So it is beta galactosidase which breaks down lactose into glucose and galactose, and glucose undergoes glycolysis. And then we have trans-acetylase. Now, lactose becomes glucose and galactose, of which glucose can undergo glycolysis, done with the help of beta galactosidase. But galactose cannot undergo glycolysis. So galactose breaks down to form acetyl coenzyme A. Galactose से acetyl coenzyme में बनता है, and trans-acetylase transfers acetyl group to beta galactosidase, and beta galactosidase will pick up acetyl from the acetyl coenzyme A. And there are lots of other metabolites. Metabolites like pyrunosidase. And pyrunosidase नाम का metabolite is produced, which is toxic to the bacteria. It is toxic to bacteria, and it is trans-acetylase which will break down pyrunosidases and break down pyrunosidases and prevent toxicity inside the bacteria. So that is the function of the three enzymes. Permease allows lactose to enter inside a cell. Beta galactosidase breaks down lactose into glucose and galactose, and trans-acetylase converts acid galactose, जो acetyl coenzyme में आया था, it will transfer acetyl group to beta galactoside, and that is done by trans-acetylase. And trans-acetylase acts on pyrunosidases, and pyrunosidases, which are toxic to the bacteria, are degraded by trans-acetylase. So this is done by all the three genes. These three genes are produced by three structural genes. Three structural genes, namely Z, Y, and A. Z, Y, and A are the three structural genes which are called LAC-Z, LAC-Y, and LAC-A genes. LAC-Z, LAC-Y, and LAC-A genes are the three structural genes. Now, when these three genes undergo transcription, they will undergo transcription to form a single RNA called as the LAC mRNA. If you remember, bacteria, prokaryotes are polycystronic. Polycystronic, which means multiple genes undergo transcription to form a single mRNA. याद है ये यूकैरियोट्स में one gene undergoes one transcription to form one mRNA which forms one protein. But bacteria prokaryotes में multiple genes undergo transcription to form a single RNA, and that is called as polycystronic. Eukaryotes, one gene, one mRNA, one protein, monocystronic. Bacteria, multiple genes, one mRNA, and multiple proteins, polycystronic. So, LAC-Z, LAC-Y, LAC-A undergo transcription to form a single RNA called as the LAC mRNA, which undergoes translation to form beta galactosidase, permease, and transacetylase. LAC-Z will form beta galactosidase. LAC-Y will form permease, and LAC-A will form transacetylase. So, LAC-Z beta galactosidase, LAC-Y permease, and LAC-Z transacetylase. Thus, three structural genes undergo transcription and translation. Now, for the structural genes to undergo transcription translation, what is required? You need the enzyme called RNA polymerase. If you remember, RNA polymerase 2 helps in formation of mRNA. So RNA polymerase 2 gets attached to the promoter gene. If you remember, जब हमने last lecture किया था transcription, where is RNA polymerase attached to the promoter gene? RNA polymerase gets attached to the promoter gene. And who picks it from the promoter gene towards the structural gene? If you remember, sigma factor. Sigma factor picks RNA polymerase from promoter gene to the structural gene, and sigma factor with RNA polymerase will do transcription. And then we have terminator gene pay. You have something called as the rho factor, which will bring back the RNA polymerase. So, sigma factor starts RNA polymerase transcription, and rho factor brings back RNA polymerase and stops RNA transcription. तो यहां पर है प्रमोटर जीन, और प्रमोटर जीन के ऊपर होता है आरएनए पॉलीमरेज. RNA polymerase, and you have O gene called operator gene. Operator gene undergoes transcription to form mRNA, undergoes translation to produce sigma factor. So, operator gene produces sigma factor. Sigma factor picks up RNA polymerase from the promoter gene and brings RNA polymerase towards the structural genes and transcribes the LAC-Z, LAC-Y, LAC-A genes to produce the LAC mRNA. So, who helps in the three structural genes to undergo transcription to form LAC mRNA? The RNA polymerase enzyme. But who will bring RNA polymerase enzyme towards the LAC-Z, LAC-Y, LAC-A gene? Sigma factor. Who produces sigma factor? operator gene. And operator gene produces sigma factor, which picks up RNA polymerase from promoter gene and brings RNA polymerase towards the structural genes, and structural genes undergo transcription to form Lac mRNA, undergo translation to form the three enzymes required for lactose metabolism. Now, what will happen? So, there is in the promoter जीन के बीचों बीच प्रमोटर जीन के बीचों बीच होता है एक जीन कॉल्ड एज द आई जीन. I gene is called as the regulator gene. I gene is called as the regulator gene. I gene or regulator gene undergoes transcription to form an RNA called as the repressor mRNA. So I gene undergoes transcription to form the repressor mRNA. Repressor mRNA undergoes translation to form a protein called as the repressor protein. So, regulator gene undergoes transcription to form repressor mRNA. Repressor mRNA undergoes translation to form the repressor protein, and repressor protein goes and binds to the operator. रिप्रेसर जाके ऑपरेटर को बाइंड हो जाता है. And once repressor binds to the operator, operator cannot undergo transcription and translation to form the sigma factor. So sigma factor cannot be produced, and there will be no sigma factor produced जब तक सिग्मा फैक्टर नहीं बनता है. RNA polymerase cannot go from the promoter gene to the structural genes. तो structural genes पे RNA polymerase नहीं आ सकता है, because to bring RNA polymerase from the promoter gene to the structural gene, you need sigma factor. And sigma factor will bring RNA polymerase to from promoter gene to structural gene to form the Lac mRNA, to form the different enzymes. But अगर ऑपरेटर जीन के ऊपर रिप्रेसर प्रोटीन आ चुका है तो सिग्मा फैक्टर नहीं बन पाएगा. नो सिग्मा फैक्टर, सो आरएनए पॉलीमरेज cannot go from the promoter gene to the structural gene and cannot produce lac mRNA. तो lac enzymes नहीं बन पाएंगे. नो लैक एंजाइम्स, तो लैक ऑपेरॉन स्विच ऑफ हो जाता है. तो सर, हाउ विल दिस बी कंट्रोल्ड? अगर एक ई कोलाई के अंदर अगर ग्लूकोज लेवल्स आर हाई एंड लैक्टोज लेवल्स आर लो, तो which is the main sugar that any bacteria will naturally break? Glucose. चाहे यूकैरियोट्स हो या प्रोकैरियोट्स हो, ग्लाइकोलाइसिस तो हमेशा ग्लूकोज का ही होता है. सो जब ग्लूकोज लेवल हाई है और लैक्टोज लेवल कम है, then at that time the repressor protein or regulatory gene produces the repressor mRNA, which forms the repressor protein, and repressor protein binds to the operator, and operator एक बार bind हो गया, it will not produce sigma factor, and sigma factor नहीं होगा. No sigma factor, then RNA polymerase cannot go from the promoter gene to the structural gene, तो lac mRNA नहीं बनेगा. And no lac mRNA, so no lac proteins. No lac mRNA, so no lac proteins. No lactose proteins. So lac operon has been switched off. Lac proteins नहीं बनेंगे. And lac operon has been switched off. So when will lac operon switch off? When glucose is high and lactose is low, then lactose is low, glucose is high. The regulatory gene undergoes transcription to form repressor mRNA. Repressor mRNA undergoes translation to form repressor protein. Repressor protein goes and binds to the operator. And operator के ऊपर repressor protein आ गया. Now repressor protein आ गया है, तो no sigma factor is produced. No sigma factor is produced. So no sigma factor, RNA polymerase cannot go from the promoter gene to the structural gene, lac Z, lac Y, lac A. No lac mRNA is produced and no lac mRNA, so no lac proteins. तो lactose proteins नहीं बन पाएंगे. So lac operon is switched off. Lac operon is switched off. Now समझो उल्टा होता है. In a cell, in this cell, glucose levels become low and lactose levels become high. तो ग्लूकोज कम हो जाता है एंड लैक्टोज हाई हो जाता है. तो रिलेटिव लेवल्स ऑफ ग्लूकोज इज लेस एंड रिलेटिव लेवल ऑफ लैक्टोज इज हाई. नाउ इफ यू रिमेंबर, there is some amount of lactose, and lactose, or you may have an isomer of lactose called as allolactose. तो lactose or allolactose, what will they do? Lactose or allolactose will act as inducer. Lactose levels are high, lactose or allolactose will act as an inducer. What will they do? The regulatory gene, I gene, produces repressor mRNA. Repressor mRNA produces repressor protein, and repressor protein के ऊपर lactose or allolactose comes and sticks to it. तो lactose or allolactose, the inducer go and get attached to the repressor protein. तो रिप्रेसर प्रोटीन के ऊपर लैक्टोज या एलोलैक्टोज आके चिपक जाते हैं, एंड दैट विल चेंज द शेप ऑफ द रिप्रेसर प्रोटीन. रिप्रेसर प्रोटीन का शेप चेंज हो जाएगा, एंड वंस द शेप ऑफ रिप्रेसर प्रोटीन इज चेंज, द रिप्रेसर प्रोटीन कैन नॉट बाइंड टू द ऑपरेटर, बिकॉज रिप्रेसर प्रोटीन का शेप चेंज हो चुका है. It cannot bind to the operator, so operator is free, and operator is free, it will produce by transcription mRNA, by translation sigma factor will carry RNA polymerase from the promoter gene to the structural genes, and structural genes will undergo transcription to form lac mRNA. Lac mRNA will form lac protein, beta galactosidase, permease, transacetylase, of which permease will allow two things to happen. Permease will allow more lactose to enter, and lactose का दो काम हो जाएगा. Lactose will keep on binding to the repressor protein and changing the shape of the repressor protein, and repressor protein cannot bind to the operator, so operator will produce sigma factor. Sigma factor will bind to the RNA polymerase. RNA polymerase will do transcription of LAC-Z, LAC-Y, LAC-A to produce LAC mRNA, which will produce more beta galactosidase, permease, transacetylase. Permease will allow more lactose to enter inside, and lactose will keep on binding to the repressor protein, and operator will allow sigma factor to carry RNA polymerase from promoter gene to the structural gene, produce lac mRNA, more permease, more beta galactosidase, more transacetylase. तो ये एक विशियस साइकिल हो जाता है. परमियस की मदद से लैक्टोज सेल के अंदर आते रहता है. Look over here. Permease keeps on allowing lactose to enter the cell, and lactose will act on repressor protein and change its shape. Because of change of shape, repressor protein cannot bind to the operator. Operator will produce sigma factor. Sigma factor will act on the RNA polymerase, transfer it from promoter gene to the structural gene, and structural genes will undergo transcription translation to form more beta galactosidase, more permease. तो ये एक vicious cycle चालू हो जाती है. And permease allows lactose to enter inside, and beta galactosidase acts on the lactose to break it into glucose and galactose, and glucose undergoes glycolysis to produce ATP, energy, and that is the meaning अभी ATPs बनने लगते हैं. So who is acting as a source of energy now? Lactose. And lactose has itself acted as the inducer and switched on the lac operon. So जब ग्लूकोज लेवल्स हाई है और लैक्टोज लेवल्स लो है, हाई ग्लूकोज will help in formation of repressor protein, bind to the operator gene, no sigma factor, RNA polymerase cannot go from promoter to structural genes, no RNA polymerase, no lac proteins, and lac operon switch off. And when lactose levels are high, lactose acts as an inducer, goes and changes the shape of the repressor protein. Repressor cannot bind to the operator. Operator produces sigma factor. Sigma factor carries RNA polymerase from promoter gene to the structural genes. Structural genes will form lac mRNA, will form beta galactosidase, permease, transacetylase. Permease will allow more entry of lactose. Lactose will act on the repressor and change the shape of the repressor. Repressor cannot bind to the operator. It will produce sigma factor, will take RNA polymerase from promoter to the three structural genes, will produce lac mRNA, will produce more permease, more lactose enters inside. Beta galactosidase breaks down lactose into glucose and galactose, of which glucose will undergo glycolysis to produce energy, and the lac operon has been switched on. So high glucose switches off lac operon, high lactose switches on lac operon. So beautifully controlled is this excellent mechanism called as the lac operon, and this is called as a negative control of transcription initiation, and an inducible operon. Lactose itself induces the operon to start, which is called as the inducible and negative control of transcription initiation. So it is an inducible operon, which is a negative control of transcription initiation, a beautiful mechanism of switching on and switching off. If you remember, we have started this lecture with genes which switch on and genes which switch off. A a protein synthesis, a gene from expressing itself. So, a gene, whether it expresses itself or whether it does not express itself. So, which gene will allow or not allow expression of LAC-Z, LAC-Y, LAC-A? इसका mRNA एंड प्रोटीन बनाने का इनहिबिट कौन करेगा? इनहिबिट करेगा रेगुलेटरी जीन which forms repressor protein which binds to the structural operator gene and does not allow Lac operon to switch on. So that is negative control done by which gene? Regulatory gene which forms repressor protein, and that is when glucose is high. And who will switch on Lac operon? Lactose itself, by acting on repressor gene, changing shape of repressor gene, and operator gene produces sigma factor, carries RNA polymerase from promoter to structural gene, produces Lac mRNA, produces Lac enzyme, and Lac operon switch on ho jata hai. So regulatory gene, by forming repressor, switches off Lac operon, regulatory gene, and internal metabolite repressor protein switches off Lac operon. Internal metabolite repressor gene switches off Lac operon, and lactose, an external metabolite, will change shape of repressor protein and switch on Lac operon. So if you remember, I made that golden statement: internal and external metabolites trigger initiation or inhibition of gene expression. So repressor protein switches off, which is an internal metabolite, and lactose switches on, which is an external metabolite.